So our research is titled Searching the Dark, Evolving Biobot Swarm Compositions to Efficiently Explore Obstructed Environments. I'm Piper. My co-authors are Caitlin Grasso, Gizem Gamuskaya, Dr. Michael Levin, and Dr. Josh Bongard. So let's unpack that title a little bit. What is a biobot? In our context, biobot is short for biological robot, and it refers to any robot that's comprised entirely out of biological components, specifically cells. Some of you might have heard of Biobot, a term coined in 2020 by Kriegman et al. Xenobots are a type of biobot, but the term biobot is cell type techniques. That's what they are, but what do they do? First of all, they move in three distinct motion patterns described as circular, linear, and curvilinear. Other than simply moving, they do behaviors like group particle aggregation. This image here shows a debris field after 24 hours of biobot motion within it. So they have aggregated the pink particles into the clumps seen in the lower right corner. So the behavior of biobots is controlled through their morphologies. One method of biobots involves the creation of ciliated spheres from stem cells, cilia being slender membrane-bound organelles that beat and cause the bots to swim through lipid mediums. When bots are produced in this way, designs can be manually manipulated onto them. So this is a video of a bot that's having a design impressed onto it by a microsurgeon. This is a very time-intensive process, and our microsurgeon cannot even drink Coke beforehand because the caffeine in that will make his hands too shaky to do it. So that's the current state of biobot, but in the future, it might not be possible to always impress AI designs onto them. This might be because in the future, biobots will be able to self-assemble or otherwise resist the impression of AI designs during their development. This video is showing some stem cells that are readhering to it. So in the future, if biobots self-assemble, then it would be possible for us, instead of to design individual morphologies, we'll design swarms of biobots. So if we self-assemble bots and we get a cohort, we could, instead of controlling them through designing individual morphologies, control them based off of who we allow to be in a swarm. So we can control them on the swarm level rather than at an individual level. So in this work, we looked into how to gain design control over a biobot collective in silico. So from a cohort of bots, how can we choose a subset from them that will lead to different behavioral outcomes? So first, we implemented what's known about the biophysics of biobots into a physics simulator. The image on the far left shows some in vitro ciliated bots. The image next to that is the body approximation we use. It's an approximation of a sphere made out of voxels. Our bots locomote based on a model of cilia that applies a force in the XY plane to each voxel on the outer edge of the bot. The image next to it shows some example forces and an example resulting trajectory. And then finally, the video on the far right shows an example motion pattern that is derived from our cilia model. So after implementing the biobot model, we chose our behavior of interest. And we chose it to be exploration as evaluated using the fractal box sampling method. So this metric encourages both large scale and fine grained exploration for the swarms. The fitness of a swarm is related to the number of boxes it visits, and the larger the size of the box, the more fitness it gains from visiting that box. This is the closed form equation of how we're evaluating swarm fitness, where n is the number of times the environment is subdivided. H is the number of boxes that a swarm visits at level n. So next, we designed some environments. We designed these environments to help understand what kinds of obstacles are easy or hard for swarms to navigate. So we designed these two environments that have a large gap between the exits of the inner cage that the bots are in. These two environments have a narrower gap between the inner cage. These two environments don't have any protrusions at the outer corners of the cage that the box are in, and these ones have these jutting protrusions. So after we combined all of these elements with an evolutionary algorithm, we use age, fitness, Pareto optimization, or AFPO. AFPO is a bi-objective evolutionary algorithm that uses age information to help search for fit and diverse solutions. We created a bot pool that our evolutionary algorithm could draw swarms from. The bot pool has 450 members, and the distribution of trajectories is approximated to what is the distribution of motion types in vitro biobots. The swarm sizes are 4 in our case, and we have 100 swarms in our evolutionary algorithm. The genome of each swarm is a vector of bots, where each element denotes the index into the set of 450 bots. So, for example, swarm 3 might be bots 2, 89, 7, and 101. Our mutation function involves swapping one biobot from our pool out for another. We implemented this by having bots that are outgoing be replaced by one with a similar motion pattern. This allows evolution to follow more of a gradient because it can be highly disruptive if like a very circular bot is replaced with a linear one or vice versa. Each swarm is simulated 6 times, where we change the starting order, and bot heading. This is to prevent the swarms from relying or exploiting their initial starting conditions to achieve high fitnesses. So these are 2 examples of 2 permutations where the bots are the same, but their orders relative to each other, their heading, the direction that they're going in is changed. And also their starting locations are changed slightly too. Okay, so that's our system. I'll talk about our results. So this is a graph of fitness over evolutionary time. The blue line is the average of top performers across several different replicates for the non-porous obstructed environment. And the shading is a confidence interval, 95% confidence interval. So if we compare these to the fitnesses of 1,500 random swarms, you see that our swarms are significantly better than randomly generated swarms for this environment. This is a similar graph for the porous obstructed environment. And again, the porous and obstructed environment, similar results. And finally, in the non-porous and obstructed environment, we also successfully gain design over swarms. So here's the fitnesses of our 4 different environments, all compared to each other over time. So the unobstructed environments, the environments without the protrusions in the outer corners, are significant. The fitnesses of the swarm are significantly higher. So those swarms are exploring significantly more area than the ones with the unobstructed, the ones with the obstruction of the corners. This indicates that the bots are more impeded by blocking things that block their way rather than trying to find a hole to get out of it. So we evolved swarms, but what behaviors are exemplary of a fit swarm? We can take a look here at an evolved and random swarm design. So anecdotally, seems like there's no very straight or very curved bots in the evolved swarms. So we did some analysis on their motion trajectories. We use a metric called curvature, which is measures how tight the circle is if they go in. A value of 1 means the bot goes in a very tight circle, where a value of 0 means it goes in a line. So there is an ideal subset of individual behaviors across each environment. And that appears to be in the curvilinear category. So The preferred bot in all environments is this intermediary curve bot. This could be due to a variety of reasons, such as increased collisions with other bots in the walls, decreased chance of getting stuck in a corner, or simply just covering more area. We also looked at the entropy of the trajectories of the bots. We used the information theoretic metric Shannon entropy. More complex trajectories lead to higher Shannon entropy. So this figure shows a deterministic and complex trajectory. Complex trajectories only occur through interactions with other members of the swarm or with the walls in the environment. So to help understand if bots were abiding by the deterministic behavior, or if they were having more complex interactions with each other in the environment, we analyzed the Shannon entropy of the curvatures for each environment. So here's the evolved compared to random Shannon entropy for the porous unobstructed environment. So it's significantly higher in the evolved swarms than in the random swarms. The same is true for the non-porous unobstructed environment, and in the non-porous obstructed environment also, and finally in our last environment too. So in the evolved swarms, if we compare the most obstructed environment, this one here to this swarm here, the Shannon entropy in the evolved swarms for this environment is significantly higher than in the least obstructed environment. So that indicates in the most constricted environment, bots have to have more complex interactions and to some degree rely on the environment to explore swarms, to explore the environment. Finally, we looked at the scalability of our solutions. We doubled the size of our swarms and had each individual bot replicated twice in the scaled up designs. We scaled up our environment designs, we doubled them in size, and we deployed both evolved and random swarms of these environments. We found that for every environment, the evolved swarm designs were still significantly better than randomly generated swarm designs. So here are videos of a random and evolved swarm design in the scaled environment. Okay, so overall, the big finding here is that we can gain design control over in silico biobot collectives without impressing individual morphologies. We also learned that some environments are more difficult to explore than others, and that our solutions scale to some degree. In our future work, we hope to do in vitro validation of our swarm designs. So we'll send our swarms to our biologist colleagues who hopefully will replicate them. The longer-term research goal is to generalize this process. We hope to incorporate more complex biophysics into our simulator so that our evolutionary algorithms can exploit collective dynamics like collisions, fusions, how bots separate. The longer-term goal of this research will create a process in which, one, some biomedical application will arise, two, the microenvironment will be replicated in our simulator, three, swarms will be evolved, and then finally, designs will be sent to biologists for in situ validation.